Good day, everyone. I'm Kishnikit Mignano. I am a second year student from BS Biology and I'm from Block 1. I am the last reporter from our group and my topic is immunohistochemistry or also known as IHC, an important diagnostic tool. Here are the content of my report. First is the information about IHC. Second is the principle. And we're going to also include the immunostaining, types of it, the methods in IHC, and its importance. So, what is immunohistochemistry? Diseases like cancer can be better diagnosed with the aid of immunohistochemistry. It can also be used to differentiate or find the difference between the various types of cancer. It is an important application of monoclonal and polyclonal antibodies to determine the tissue distribution of an antigen of interest in health and diseases. Let's come now to the contribution of immunohistochemistry in pathology and neuropathology. In pathology, it is widely used in diagnostic surgical pathology. All anatomical and surgical pathologists use IHC to confirm cancer cell type and possible origin of metastatic cancer of a non-party site. And in neuropathology, it plays a big role, especially in diagnostic neuropathology. Why? Because the discovery of immunohistochemistry helps the neuropathologist to be able to distinguish the specific antigenic differences between the cell and the infectious agents or pathological cellular inclusions. Let's come now to the principles of immunohistochemistry. As you can see in my PowerPoint, I only put the keywords because if I put the whole principle, it would be too long. So let me just discuss it to you guys. So allow me to speak in Taglish so that I can explain it briefly or clearly to you guys so that you can all understand clearly. So the principles of IHC was existent since 1930, but the first study about IHC was reported in 1941. So, Guns, kasama yung mga katrabaho niya, they use the fluorescent isothiocyanate or also known as FITC na they are labeled antibodies that have a fluorescent dye to localize the pneumococcal antigens in infected tissues. So, sa pamamagitan ng pag-expand at pag-develop ng IHC technique, na-discover natin ang tinatawag na enzyme labels. Ito naman, ang mga halimbawa nito ay ang peroxidase at alkaline phosphatase. Tapos, na-introduce sa atin ang colloidal gold levels. Ito naman ay ginagamit para ma-identify ang immunohistochemical reaction sa pamamagitan ng paggamit ng both light at electron microscopy levels. Tapos, meron po tayong mga ibang mga labels. Ito ay ang radioactive elements at saka immunoreaction elements. Ito naman, sila mabivisualize mo sa paggamit ng autoradiography. Ang pinaka-aim naman ng IHC ay ang pag-perform ng IHC staining sa mga pasyente pero konti lang ang madadamage ng mga cell or tissue. Tapos, makikita mo na yung mga tissue typing or tissue markings, pero konti lang ang madadamage or masisira ng mga antibodies. So, ano nga ba ang immunostaining? Sa biochemistry, ang term na immunostaining ay nag-refer sa isang kahit anong application ng mga antibody-based method para makadetect ng protein sa isang sample. Tapos naman, ginagamit din ito sa pag-detect ng isang specific protein maker na makakatulong sa atin magkaroon ng accurate na tumor classification at magkaroon tayo ng accurate diagnosis. So, bakit natin ginagawa ang immunostaining? Ang immunostaining ay ginagamit sa mga cell biology para mag-study ng mga different protein, localization, at saka distribution sa isang tissue, cellular, at saka subcellular. We have types of immunohistochemistry. We have the enzyme-linked immunosorbent assay. We have flow cytometry. Also, the immunoelectron microscopy and the Western blotting. The first type is the enzyme-linked immunosorbent assay. ELISA uses a solid phase type of enzyme immunoassay to detect the presence of ligand in a liquid sample using the antibodies directed against the protein to be measured. The next one is the flow cytometry. It is a lab test used to analyze characteristics of a cell or particles. During the process, a sample of cells or particles was suspended in a liquid and injected to the flow of cytometer machine. The third type is the immunoelectron microscopy. It is a method used in electron microscopy to detect or diagnose a viral infection. The last type is the Western blotting. This method involves using a gel electrophorese to separate the proteins. The separated proteins was transferred out of the gel to the surface of the membrane. We come now to the method of immunohistochemistry, and they are one-step direct method, ABC method, two-step indirect method, and a pyramid signal amplification. The first method is the one-step direct method. This method consists of using a labeled antibody such as FITC conjugated with an antiserum, enzyme, or alkaline phosphate that reacts directly with a specific antigen in a tissue section. The next one is the ABC method. It is a three-step detection method. The primary antibody binds to the antigen in the tissues, and then it is in turn to recognized by the biotinylated secondary antibodies. Complex complexes are formed or can be formed by avoiding, and biotinylated enzymes can be attached to the latter. The third method is the two-step indirect method. This method utilizes a unlabeled primary antibody to detect the antigen of interest in the tissue, and then the secondary labeled antibody used to bind to the primary antibody. And the last one is the pyramid signal amplification, or sometimes known as capitalized reporter deposition. It is a highly sensitive method enabling the detection of a low abundance target in fluorescent immunocytochemistry and immunohistochemistry. And the last one is the cytohybridization applications. This importance means na malaki talaga ang naging contribution ng IHC sa medical field at sa research field. Dahil doon, sa medical field naman, dahil na-invent ng IHC, nagkaroon ng capacity ang mga, or capability ang mga doctors na ma-identify at mabigyan ng proper treatment ang isang cancer patient. So, in the research field naman, nakatulong ito sa mga researchers na nag-study ng study about cancer. Kasi mas ngayon, kaya na i-identify kung anong cancer cell ito. And nakakapag-create sila ng mga treatment or mga ways on how to treat a cancer patient. So as you can see, the IHC has really big contribution, especially in our medical field and research field. This is the summary of my report. This is the summarization of my whole report, and you can see here, you can read here the main point of my study. And the next one, the references. These are the references where I got my ideas about my report for today. And the next one, I was given the honor to introduce to you my group members. They are first Falcolan Stefan Ashley, Malarta Jama Chris, Lapuz Mary Alea Grace, Castro Alexa May, and yours truly, Minyana Krishniki. So thank you again from my protein molecules to yours. I sincerely thank you guys and have a good day.